Pharmapal.
The role of Anastatica Hierochuntica methanolic extract. Welcome to our channel. Today, we're diving into the fascinating world of Anastatica Hierochuntica, a plant with a rich history in traditional medicine. We'll explore its potential as a natural remedy against acute myeloid leukemia, AML. Recent studies reveal that its methanolic extract shows significant cytotoxic effects on AML cells while sparing normal human cells. Join us as we unpack the science behind this remarkable plant. Let's kick things off with a brief introduction. Anastatica Hierochuntica has been used in Egyptian folk medicine for various ailments, including gastrointestinal disorders and diabetes. However, scientific research on its medicinal properties has been limited. Our study aimed to investigate its cytotoxicity, that's the ability to kill cells, specifically on AML cells, a type of cancer that affects the blood and bone marrow. Now, let's talk about how we conducted our research. We used the following materials and methods. 1. Plant material. Dried aerial parts of Anastatica Hierochuntica were sourced and prepared for extraction. 2. Extraction process. The plant was ground and defatted, then extracted with 70% methanol to obtain a concentrated extract. 3. Cell lines. We tested the extract on isolated AML cells and normal human peripheral leukocytes, NHPL. 4. Cytotoxicity assessment. Using various assays, we measured the extract's effects on cell viability and apoptosis, which is the process of programmed cell death. This systematic approach allowed us to gather reliable data on the plant's potential therapeutic effects. So, what did we find? The cytotoxicity of the methanolic extract on AML cells was significant, with an IC50 value of 0.38 g/ml. This means that a very small amount of the extract can effectively kill half of the cancer cells in our tests. Importantly, the extract showed non-significant cytotoxicity on normal cells, indicating a level of selectivity that is crucial for any potential treatment. We also observed changes in key proteins involved in apoptosis. Specifically, there was an increase in caspase-3 levels and a higher BAX/BCL2 ratio, which suggests that the extract induces cell death through the mitochondrial pathway, independent of the p53 protein, a well-known tumor suppressor. The ability of Anastatica Hierochuntica to selectively target AML cells while sparing normal cells is promising. This suggests that it could be developed into a natural anti-cancer agent, potentially leading to new treatment options for AML patients. Moreover, the mechanisms we uncovered highlight the importance of further research into the bioactive compounds present in the plant. By understanding how these compounds work at a molecular level, we could enhance their effectiveness and possibly combine them with existing therapies for even better outcomes. Next, let's discuss the cell viability assessment. The study employed a dual staining technique using an XNV and propydium (PI) to differentiate between viable, apoptotic, and necrotic cells. Viable cells, these cells do not stain with either dye. Apoptotic cells stain positively with an XNV but negatively with PI. Necrotic cells stain positively with PI. The researchers evaluated approximately 1,000 cells per slide, providing a robust dataset for analysis. Immunocytochemical study. Next, we move on to the immunocytochemical study of AML cells. The cells were smeared on positively charged slides and treated with specific antibodies to assess the levels of key proteins involved in apoptosis, including p53, caspase-3, BAX, BCL2. Using an avidin biotin complex immunoperoxidase technique, the researchers scored at least 5 fields per stained slide, calculating the mean percentage of positive cells. This scoring system allowed them to quantify the expression levels of these proteins effectively. Quantitative real-time PCR. Now, let's talk about the quantitative real-time polymerase chain reaction, QRT-PCR. This technique was used to measure the expression of apoptosis-related genes, such as p53, caspase-3, BAX, and BCL2. RNA isolation. The total RNA was isolated using the RNA plus mini kit. cDNA synthesis. Complementary DNA was synthesized, which is crucial for the QRT-PCR process. The expression values were normalized to GAPDH, a common housekeeping gene, ensuring reliable results. Statistical analysis. Moving on to statistical analysis, All experiments were performed in triplicate, and the data were presented as mean plus or minus standard deviation. The researchers utilized one-way ANOVA or student's t-test to analyze the data, considering a probability level of p less than 0.05 as statistically significant. Now, let's summarize some of the key results illustrated in the figures. 1. Figure 6. This figure showcases the qualitative determination of cell death stages using an X and V/PI dual fluorescence staining. The results indicated that dead cells were stained orange to red, highlighting necrotic or late apoptotic stages. 2. Figure 7. Here, we see the immunocytochemical reactivity of apoptosis-related proteins. The study found a statistically significant increase in Caspase-3 protein levels and the BAX/BCL2 ratio in treated cells compared to controls. What does the dual fluorescence staining with an X and V and PI reveal about the mode of cell death in AML cells? The dual fluorescence staining technique using an X and V and propydium iodide allows us to distinguish between viable, apoptotic, and necrotic cells. In our study, cells stained positive for an X and V and PI indicate late apoptotic or necrotic stages, while viable cells remain unstained. This method revealed that after treatment, we observed a significant increase in the number of late apoptotic or necrotic cells, suggesting that the extract induces cell death primarily through apoptosis and necrosis. How did the expression levels of apoptosis-related proteins change in response to treatment with Hierochuntica? The immunocytochemical analysis showed a statistically significant increase in Caspase-3 levels, approximately 2.5 to 3.2-fold compared to untreated controls. Additionally, the BAX/BCL2 ratio increased significantly, indicating a shift towards pro-apoptotic signaling. However, p53 expression remained unchanged, suggesting that the extract may induce apoptosis through a p53-independent pathway. What role does the BAX/BCL2 ratio play in determining the fate of AML cells? The BAX/BCL2 ratio is crucial in regulating apoptosis. A higher ratio indicates a pro-apoptotic environment, promoting cell death. In our findings, the significant elevation of this ratio in treated cells suggests that Hierochuntica enhances apoptotic signaling, leading to increased cell death in AML cells. This shift underscores the extract's potential as a therapeutic agent. What implications do these findings have for the treatment of AML? The results indicate that Hierochuntica has the potential to induce apoptosis in AML cells, which is significant given the challenge of chemotherapy resistance in AML treatment. By targeting apoptosis pathways, this extract could serve as a complementary therapy alongside conventional treatments, potentially improving patient outcomes. How did the study ensure the reliability of the data obtained? The study employed rigorous methodologies, including using control groups, random field selection for imaging, and analyzing at least 5 fields per stained slide. Statistical analyses were conducted using one-way ANOVA or student's t-test, with a significance level set at p less than 0.05, ensuring the robustness of the findings. In summary, the study presents compelling evidence for the pro-apoptotic effects of Hierochuntica on AML cells. The dual fluorescence staining and immunocytochemical analyses provide insights into the mechanisms of cell death, highlighting the extract's potential as a therapeutic agent. Thank you for your attention, and I look forward to your thoughts and discussions on these findings. Thank you for joining us today. If you found this video helpful, please like, subscribe, and hit the notification bell for more content like this. If you have any questions or comments, feel free to leave them below. Until next time, keep exploring the wonders of science.
Pharmapal.